I'll introduce the next speaker. I was interested watching John's presentation and him acknowledging one of his mentors, and I'd introduce the next speaker in a similar vein. I think having had the good fortune to work at the Duluth Lab, virtually everybody who works there has been a mentor to me, because it touches on something Joe said. It's really a huge advantage if you are doing your work and you run into a question that in that environment you can usually walk down, whatever the question that comes to your mind, you can usually walk down the hall and engage in a discussion with somebody who's got a lot of insight. But there have also been some real important external mentors to me in my career, and I'm fortunate enough to introduce one of those, who next to my graduate major advisors are probably one of the most influential people in my career. And I would say point to two things. One is the interactions I've had with Dominic have taught me to look for the larger picture, the theories that connect it all, whether they're true or not, right? and also that the perfect is the enemy of the good, and to not get too focused on the exceptions. And we are studying larger principles, and the challenge is sometimes to see those. So I'm pleased to introduce Dom DeToro of the University of Delaware.
You know, it goes both ways. I was trained as an electrical engineer, if you're wondering where I came from. And in the first 10 years of my life, I spent modeling phytoplankton and toxic chemicals in surface waters. And then in 1980, Norb Jaworski became the director of the Duluth laboratory, and he needed a pet engineer to help him out, talking to all of the toxicologists. And he turned to my direction and invited me up there. And the Duluth laboratory was my postdoctoral training in toxicology, and most of the work that I've done following basically came from interacting with all the people at the Duluth lab. So you should understand that this talk is basically how they influenced me, and then to some extent vice versa, but it started with them influencing me, not the other way around. One of the advantages, by the way, of not being professionally trained in a subject is you don't carry all the biases. So no one ever told me you couldn't do this. And so a few things that I did, I just did because I didn't know enough to know that it was too complicated. So for young folks, forget all the classical stuff. Most of it's wrong anyway. This is the keynote slide in the talk I'm going to give you. This is a paper that Gil Weiss published in 1983, just early on in my, as I say, in my postdoctoral career at Duluth. And what he plotted here was the LC50, 4-day LC50 on fathead minnows, versus the octanol water partition coefficient. Each one of those dots is an individual chemical. And they all line up. It is remarkable when you see something like that. If you see something like that in a talk you're listening to, pay attention. There's a career there to be made. First question. How in the world does it happen? How in the world do all the chemicals line up like that? It's astonishing. This is fish mortality, and this is just a hydrophobicity indicator. So something's going on. Something fundamental is going on, point one. Point two, this is the aqueous solubility. And interestingly enough, when you get certain chemicals, they're not soluble enough to deliver enough. Well, now we know they're not soluble enough to deliver enough material to kill the organism. So that's where there's a cutoff. And the third thing is that this is data generated by the Duluth lab themselves, fathead minnow LC50s. I asked Don Mount earlier on when they decided to actually build a chemical laboratory that was associated with the toxicological group, and that was critical to understanding their own data because you had chemists that could put it together and you had a lot of data. And I'm from the datavore community. We eat data in large quantities, so you need to have a cooperation with a laboratory like Duluth. So how does it work? What's going on? Well, the first people to take a look at it was actually McCarty and Mackay. Lyle McCarty and Don Mackay took a look at it and had an interpretation of what was actually happening based on the BCF models that Deb Swackhammer presented before. I will show you a simpler way of understanding it. So remember the question, what's going on? Why is this working? So let's imagine first that the octanol water partition coefficient approximates. This is a very annoying computer. This is a Mac? No, it isn't. that the octanol water partition coefficient approximates the lipid water partition coefficient. We'll assume that lipid is the site of action for this toxicity. Furthermore, if you imagine a chemical whose octanol water partition coefficient equals 1, not 10 to the 1, 1, and the lipid water partition coefficient then would also be 1, then if you go in here for that chemical and read over, you would get the lipid concentration at the LC50, because this compound, the same concentration in water, is the same concentration in lipids. So that is the concentration in the organism that caused the 50% mortality. You see? So you can get the critical body burden just by that simple extrapolation. You might remember, you probably don't, but once Veith published that paper, there was a cottage industry that developed of publishing plots where you had slopes and intercepts of various organisms and various classes of compounds and so on. So this gave us what the intercept meant. But what did the slope mean? What was that about? So this is where we picked it up. The first thing I'd like you to notice, so this is a plot that we put together where we made the following assumption. Let's assume that the critical body burden is the lipid water partition coefficient times the LC50, previous slide. And let's assume also that, well, if you take the logarithm of that, you get the critical body burden and you get the lipid water partition coefficient. So now we're going to build a QSAR for the lipid water partition coefficient, and it's just a constant times the octanol water partition coefficient. So now what we're doing is relating chemical property to a physiological property. What is the partition coefficient? So if you then make that substitution, you get the critical body burden as the intercept, and then you get a slope to the octanol water partition coefficient. That's the slope. And the critical assumption we made was that the intercept was not species dependent. The partitioning to the lipid, to the toxic, the toxic mode of the place, the target lipid model is what I'm trying to remember. We named it target lipid because we didn't really know what was going on, but we knew that something must have partitioned. And so that was the assumption, that A, the narcosis slope was a constant across all organisms. Why not? If you have a slope that depends on every organism, then you need a BCF model for every organism, and you haven't gotten anywhere. So that was the assumption. So this is all the data that we put together, and it comes from all of the EPA, from the Duluth laboratory assemblage of all the data. And what I've done in this paper, by the way, is when you see red, that's a Duluth lab contribution. So the Ecotox is now all the toxicological data. Smiles, Dave Weininger, when he's there, invented that. So the way we got octanol water partition coefficients is to just queue in the smile string and get it from, and then also, look, this is the fathead minnow database, an enormously large database. One of the things that's useful, by the way, when you make the constant slope assumption, you see this data sitting way out here? You can extrapolate that all the way back and get the critical body burden because you know the slope with high certainty because there's thousands of data points that determine that slope. So this was the contribution, and this was the beginning of the narcosis model that we used. It's interesting. So, okay, we've got LC50s for a whole bunch of organisms. What about acute to chronic ratios? Well, if you look at narcosis and you just cross-plot the acute LC50 to the chronic for a whole bunch of organisms, remarkably, that's a slope 1 line. You get an ACR of about 5, quite constant across many organisms. And the ACR, of course, was a great contribution that Don Mount and Chuck Steffens made. Think about that assumption, that there's a relationship between the acute criteria, mortality, and the chronic non-mortality effects, long exposures, different modes of action. That takes courage. That's a lot of courage, and that's still used to give you some idea of how powerful that assumption is. And so you can see that the thing is, this is one annoying computer. You can see how powerful it is. Fish, crustacea, insects, no problem. Secondly, since you've got critical body burdens, you can do a species sensitivity distribution. You just plot them on a probability plot, and you go down to the 5th percentile using Russ Erickson's extrapolation. And bingo, you've got the final acute value. So you can use this narcosis model to actually put it in a criteria framework. Another great contribution from the EPA labs and Duluth in particular. The current status, by the way, of the amount of data that we have is quite large. There are over 1,000 data points. There are I think 40 or 50 species and so on. Next. we needed, the next contribution was, okay, I've got one chemical that's a narcotic. Suppose I've got two chemicals that are narcotics that are mixed up. What's the toxicity when you mix them? So Broderius cooked up the so-called isoball plot, and this is an example of that. You plot the toxicity of he used 1-octanol, just to confuse everyone. He used 1-octanol as the narcotic baseline. It's an alcohol, and that's why he did it. And then the second toxicant, and if they follow a straight line, suitably normalized, then they are additive. Well, that turns out to be very important because that suggested that perhaps narcosis, chemicals that all operate by the same mode of action, are additive. And some experiments that were done, boy, this is annoying. Some experiments that were done, for example, the 14-day LC50 guppy experiments by Koneman, who published the plot that you saw initially that Weitz published at about the same time. He took 50 compounds at 1/50th of their LC50. So that's from micrograms per liter all the way up to tens of milligrams per liter. 1/50th, mixed them all together, and then use that batch to get an LC50. We would have predicted 9/10 of one toxic unit. He found experimentally 9/10. 50 compounds, experimentally additive. So it's clear that narcotic chemicals add up. Just to give you a brief, what's a toxic unit? What you do is you take the concentration and you divide it by the LC50 for that chemical for that organism. Well, it turns out, and then the total toxic units are just the sum of the toxic units of each of the chemicals in the mixture. Well, it turns out it's relatively easy to show that toxic units are additive because the critical body burdens are additive, because toxicity doesn't depend on the nature of the compound. Narcotic toxicity is independent of the properties of the compound, only KOW, and that's just the partitioning into the organism. So it doesn't matter what compound is piling up. They're just additive. It's actually a separation. It's the molar volume that appears to be doing it. And Don Mackay actually tried correcting everything for the molar volume. It doesn't matter too much. It's only less than a factor of 2. So the narcosis model actually tells you that. But perhaps so why would we develop this? Well, we developed this because as I was doing my postdoc at the university, at the Duluth laboratories, sediment criteria had to be developed for PAHs, and certainly PAHs are mixtures. And we had sediment criteria for each of the PAHs, but then we had to put it all together. And the question was, how are we going to do it? And the genesis of that was, and in fact, Dave Mount and friends were central to. One of the nice things about having a postdoc at the Duluth lab is you can suggest experiments, and then they have this wonderful experimental facility and generate lots of data to confuse us for a while, and then we straighten it all out. It's been a wonderful experience, let me tell you. Finally, by the way, narcosis is not the only mode of action. McKim and Company published, Weitz and Broderius, by the way, published a paper where they showed that there was another class of compounds, which are now called polar narcotics, which seem to be more toxic. This is LC50 versus KOW. And you can see that the LC50s are lower. We think that actually occurs because the partition coefficient is not related to octanol water partition. The lipid-water partition coefficient for polar compounds doesn't work very well for PAHs, for polar compounds. And finally, the modes of action in general have to be started to be, and that's a current active area of research. Not everything's a narcotic. Everything is a narcotic if it doesn't kill by some other mechanism, but they're much more sensitive compounds, of course. So where are we going? When I give a longer talk on this subject, I declare the end of the age of octanol. We are done with octanol water partition coefficients. They work beautifully for the compounds we first had to deal with. Luckily, think of where we'd be if we didn't have octanol water partition coefficient initially. We'd still have we wouldn't have very much, but we are now in the world of much more complicated compounds. And what's happening now is we're using models that are actually much more sensitive to the details of the chemistry. And so the partition coefficients are no longer just A1 times the log of the octanol water partition coefficients. They are bilinear regressions where the solvent water system are the lowercase letters. So if you're interested in lipid water, you have to generate the lowercase letters. And then each of the solutes are the uppercase letters. But the nice thing about it is that for every solvent and solute, you don't have to do both individually. You can use the solute parameters once you've got them for any of the Abraham relationships. And so this is where it's going. And this is the Narcosis data, predicted versus observed for quite a lot of chemicals and quite a lot of data points. This plot, by the way, is a bivariate histogram. The number of data points in each of the little diamonds is indicated by the color coding. So instead of getting a blob, you can actually see what's going on. So we play around with that. And with that, I would like to thank my colleagues and friends at the Duluth Lab. It's been a wonderful experience. Thank you.
Questions for Dominic? Or other comments?
So I finally realized, as have maybe many others who have watched your brilliant presentations over the years, that part of the reason why they're so good is fundamentally you only have one presentation that you just give over and over again. They all look the same. It's those all log plots in there. So what I was wondering is, so one of the reasons why working with you has been so much fun is because you have helped us put together engineering and biology and chemistry and all this sort of stuff, and we do that. So I wonder what advice you would have for young graduate students or early career people as to how to get involved in those kinds of problems.
The question is, for the younger students in the crowd that are embarking on a career, how should they approach learning enough material so that, my first suggestion would be to take a postgraduate position at the Duluth Laboratory, which is the way I learned it. The second thing is to make sure that when you're in college or in graduate school that you take hard courses. You don't need a course in environmental awareness. You need a course in organic chemistry, or physical chemistry, or mathematical modeling, or it's important that you broaden your horizons. And the other thing is, most of what I've done, I've taught myself. So the other thing you have to learn how to do is teach yourself new problems. As John Nichols was saying, he didn't know anything about PBTK modeling, and he taught himself it. It's possible. One of my colleagues once said to me that one of the most important things that he learned when he worked with us was not to be afraid of a new subject. If other people can do it, you can do it. The hard part is inventing it, but once it's been invented, you can learn it relatively straightforwardly. So don't be afraid of the new topics. Inventing things are hard, but using other things that people have invented and sort of shifting them over to where you need them is relatively more straightforward. And the third thing I would advise you to do is to take 3 courses in thermodynamics, because the first course you don't understand anything, and the second course you're beginning to get some idea what's going on, but the third course, and take it from the chemical engineers, is where you really learn what's going on.
Anybody else have a way to keep Dominic talking?
I didn't know that was a problem.
All right, we'll just bring it.
Thank you.
So I'll let the people shuffle in. Thanks, Dominic.